So today we are going to look at ammonia. Basically the transamination process would be dealt over here. First we'll look at the sources from where we get ammonia. Amino acids and nitrogen compounds are two things which basically contain ammonia. Now ammonia exists in NH4 plus form, that is ammonium form, and this form which is present maintains the acid and base balance in our body. Ammonia is said to be highly toxic in nature. Now we'll look at transamination process in detail, and in transamination first we'll look where does it occur, after that which enzymes are used, and the entire overview of the cycle is also important. So transamination occurs basically in the cytoplasm of cells. Now it occurs in cytoplasm of all the cells. So it could be liver cell, it could be in the muscles, it could be in any organ. In transamination there is amino acid 1, along with that there is keto acid 1. It is getting converted into amino acid 2 and keto acid 2. What does it indicate? The definition here is much more important. Here there is exchange of amino group, that is NH2 group, okay, from the first amino acid, okay, and it goes towards the keto group of the next amino acid to form a new amino acid, right? It gets transferred to the new amino acid formation. It's between one amino acid and keto acid. Now if we look ahead, this entire process is taking place just for the formation of a new amino acid from an old amino acid. It's nothing new and old, but just this transfer of amino group. So this is transamination. Now we'll look at the basic overview, what actually takes place, why is NH2 group transferred, and why these processes are occurring. All cells which are present in our body, they produce amino acid 1 to 2, like that. Transamination occurs. Now this amino acid 2 which is there, it is mostly glutamate, which is said to be produced as glutamate. Glutamate has to be transferred to liver. Now why does it need to be transferred to the liver? There is a certain enzyme which is known as glutamate dehydrogenase. Now glutamate dehydrogenase is the enzyme present only in the liver. So only liver has the enzyme glutamate dehydrogenase. For that purpose we need to transfer the amino acid 2 to the liver. So now how would we do so? We'll convert the glutamate which is formed, okay, glutamate is also known as glutamic acid, both are the same. We are going to convert it into a much better form, that is glutamine. This is done by the addition of ammonia group. Glutamine which is there, it is going to house, or it is storing the ammonia which is present, and it is going to take it to the liver. Now when it reaches the liver, the glutamine is just going to undergo breakdown with the help of glutaminase to form glutamate again. So nothing happened, just glutamate was converted to glutamine to form glutamate again in the liver. Now it undergoes oxidative deamination. So first transamination, now oxidative deamination. This results in the release of ammonia, okay. Now when ammonia is released in the liver, it undergoes a cycle which is known as urea cycle. Urea cycle is used to produce urea from ammonia. Because ammonia is said to be toxic in nature, so our body needs to convert ammonia to urea to be excreted out. So this was entire overview why transamination, oxidative deamination and urea cycle occur in our entire system. So after the entire overview, we'll go to transamination process. We saw transamination occurs in all the cells of our body, right? So it can occur with the help of certain enzymes, the common enzyme, that is transaminases, which can also be called by another name, that is amino transferases, which transfer amino group, okay, amino transferases, that's not amino transaminase. If we move ahead, there is certain enzyme, there's certain prosthetic group attached to this enzyme, that is PLP, that is pyridoxal phosphate is the prosthetic group. And if we look at PLP, it is said to be derived from vitamin B6. This is important because when we come towards the chapter of vitamins in biochemistry, here we need to know that where is this vitamin used and in which form it is used in certain cycles. After that, transamination process is a reversible process, and this reaction can be reversed. Okay, we would look at an example for this. First, in this example, we'll take alanine. Alanine is said to be converted to pyruvate, and with the help of alanine transaminase, which is also written in the form of SGPT. It forms pyruvate. Now what happens over here is there is transfer of amino group taking place from pyridoxal phosphate to pyridoxamine to form pyridoxamine, pyridoxine phosphate. Okay. Now this pyridoxine phosphate transfers the amino group which it has taken from alanine, okay, to alpha ketoglutaric acid. Now what happened over here is we are forming a pyruvate as the keto acid which is formed from alanine. Now alpha ketoglutarate acid is the second keto acid which is present in the reactant which is going to form an amino acid. That is, obviously we said glutamate is going to be formed in most of the reactions, so your glutamate would be formed. So what took place over here is two products are formed, pyruvate and glutamate. The enzyme in this process is also alanine transaminase or SGPT. So the two products which are formed over here are glutamate, pyruvate. Now if we want to know how the products are formed, we'll write the steps down in detail so that we would understand it. Here first what happens, there is transfer of amino group. Because this is transamination, so amino group is transferred from the alanine part to the PLP part, that is pyridoxal phosphate, which goes to pyridoxine phosphate. Then it goes to alpha ketoglutarate. And from alpha ketoglutarate is the keto acid. It goes to the amino acid, right, which is the glutamate. Now the glutamate which is formed now is the product along with pyruvate. Now if we look at another example where we take aspartate along with alpha ketoglutarate. This aspartate and alpha ketoglutarate in the presence of GDH, where GDH stands for AST, that is aspartate transaminase or SGOT, in the presence of pyridoxal phosphate gives oxaloacetate and glutamate. Again we got glutamate, so all these glutamate are going to be transferred from our body cells to the liver. Now one thing we need to know over here is there are certain exceptions. Certain amino acid does not undergo transamination, rather they directly undergo deamination, and that too non-oxidative deamination. They include lysine, threonine, proline and hydroxyproline. These need to be remembered. Lysine, threonine, proline and hydroxyproline. They undergo non-oxidative deamination. After that, there is certain delta amino acid which also goes this transamination process, that is ornithine. So ornithine is the delta amino acid which shows transamination process.